Hello everyone, welcome to another lecture series in Basics of Virology. I'm Dr. Lokesh Gambhir. So, in past few lectures, we have understood multiple things about viruses, the mode of infection of different viruses, how the replication cycle is going about, what is establishment, what do you mean by the establishment of antiviral state, what are the immune responses that are being generated against the viruses, and we have looked at certain critical examples, including HIV also. So today, I have come up with another major virus which one should be aware in terms of understanding the biology and immunology part. See, we are going for this lecture series simply because we as a SGVU, Suresh Gyan Vihar University family and the applied science family firmly believes that we have to bridge this gap. What gap? We have to bring the academics a click away from every student. Click away from every student. You just need to click on your YouTube channel, and then you'll be getting the eminent speakers giving the deliberations with on different topics. So that's the whole idea. That's the whole idea of creating that e-learning center. So today, in under the ages of e-learning center, I'm bringing you a topic which is very much relevant in terms of Indian scenario also, the topic of dengue virus. Now, the word is nothing new, right? In India, we every year we hear that yes, in this area dengue is there, in this area dengue is there. People are suffering from dengue fever, and then there's certain mortalities that are associated with this particular dengue fever. So it's very important for us to first of all understand what is dengue virus, what is dengue. The dengue is corresponding to the disease that is being caused by dengue virus, and then if you're talking about dengue, then what are the signs, what are the WHO guidelines, how you categorize the categorically separate the symptoms, and we will be understanding that what exactly is dengue virus in terms of structure, in terms of replication cycles, what are the antiviral targets. So the main objective of this particular lecture, the main objective of this particular lecture is to impart an understanding, what is actually dengue virus, what is the structure of this particular virus, how this virus causes immune evasion, and the transmission mode, what is the transmission mode of this particular virus. So by the end of the lecture, I expect you to understand all these things in a single lecture scenario. So without any further delay, let us go directly to the topic. So see, when I'm talking about dengue virus, it belongs to a family called as Flavivirus. Now Flavivirus or Flavivirus, see, it's a very huge family. It's a very huge family. It contains 65 different types of viruses. So all those viruses are enveloped viruses, and they're enveloped in enveloped glycoprotein viruses. But they share a very common antigens on their surface. That's why it's very easy to diagnose them or categorically put them into a single family of Flavividae. But the main point, what I want to tell you is that when I'm talking about 65 different viruses in a single family, this is the family whose one third of the members are pathogenic to humans. They are pathogenic to humans. So almost above 20 viruses in this family are there, which are pathogenic to humans, like dengue virus, like West Nile virus. So that's why this particular family has been in the hotspot of virology or for the investigations of different for deciphering the replication cycle of the human pathogens, so that we could have a putative treatment and the therapy module against the infections of those particular viruses. So what are the general features when we're talking about in terms of flavivirus? See, when I'm talking about the general feature in multiple lectures, I'm talking about the general features of a certain family. Please have a clear cut understanding that when we are discussing the general features, we are talking about a family. Now in that family, if suppose there are 4 members, so I'm talking about the general features based on that family, what behavior these 4 will be having. Yes, but that doesn't mean that they will not be having their individual identity. Yes, they will be having the differences among them, they will be having their individual identity, but there will be certain similarity because they belong to the same family. And that is that the members of these viruses replicate in the cytoplasm. They never go inside the nucleus, they will always be replicating in the cytoplasm. And then they followed the cell network, cell trafficking system, the cell secretory pathway, the pathway that goes from endoplasmic reticulum to Golgi body to plasma membrane, and then they are released out of the cell. So they follow that path to go out of the cell. Once the mature virion is available, then the mature virion follows this path and goes out of the cell. And not only that, see, all the family members and the Flaviviridae, as per the Baltimore classification, we have categorized them as a single strand positive sense RNA viruses. So that positive sense RNA as a genome behaves as a putative mRNA, which can directly give rise to protein. And while replication, if you are talking about the replication of a plus sense RNA, then the RNA polymerase will be using this plus sense to make a negative sense. So that particular negative sense is called as antisense. So it has the potential to convert the mRNA into antisense. And then that antisense is used as a template to create the multiple copies of the original template. And that's how the replica of the original templates are generated. So you see, there is a transient stage. There's a transient state, or I should say the intermediary stage, which gives rise to another single strand, which is complemented to the plus strand, and it's called as antisense strand. So this is of course the general feature, right? This is of course the general feature, and this is the feature of all the kinds of plus 10 RNA viruses, plus sense single strand RNA viruses, because that's how the replication will be proceeding. So when we are talking about the multiplication, yes, we do discuss certain common features, such as general features. When we are talking about the clinical manifestation, then in this particular family there are multiple viruses, as I showed you, as I told you. And then among those group of viruses, if we are talking about the main limelight diseases that we are talking about in terms of clinical settings, and I'll be talking about St. Louis encephalitis, Japanese encephalitis, tick-borne encephalitis. Of course, dengue is one of the major causes of mortality based on this particular viruses. Then hemorrhagic fever is there, caused by forest virus. Then we are talking about the hemorrhagic fever that is induced by yellow fever virus, which also includes hepatitis in between. So this, all the preamble that I had given you, is regarding the Flaviviridi family or the Flaviviruses. So right now we'll be picking one human pathogen from that 65 different types of viruses that are belonging to the family, and with the hope to deliberate and to explain this particular example to its full potential, so that you may understand that based on this example of dengue virus, how the other 64 types will be behaving. Although I told you that they will be having their individual identity. So maybe in forthcoming lectures we'll be looking at some of the other examples of this particular family. But for right now, let us focus and let us try to understand what exactly is dengue virus. See, dengue virus, the nomenclature, how it came. See, when we are talking about the virus nomenclature, we already understand that it's given by a certain scientific committee. Yeah, it's okay. But what is what is the basis of giving a name? How you pronounce a name? See, you give the nomenclature to the viruses based on different attributes, depending on the host. It's an animal virus, it's a plant virus. Depending upon the disease, immunodeficiency virus, HIV, hepatitis virus. Depending upon the disease, you are giving a nomenclature to the virus, and that is the attribute that was picked up in naming this particular dengue virus. Why? Because it causes dengue. That's why I'm calling it as dengue virus. Fine. And then this particular dengue virus induces dengue, and I have written another term, that is DHF. We call it clinically as DHF, that is dengue hemorrhagic fever, which is the severe case of this particular dengue, that we'll be discussing in the forthcoming slides. And then these viruses are arboviruses. Arboviruses means they are transmitted by an intermediary host or intermediary vector, and that vector is of course mosquito. So the viruses which are transmitted from mosquitoes are called as arboviruses. And as I told you that it is a single strand RNA. This single strand RNA dengue virus has 4 serotypes. Dan 1, 2, 3, 4. Dan 1, 2, 3, 4. 4 serotypes. Now, what are serotypes? Majority of us have a very clear understanding what are serotypes. These are strains in a species which can be distinguished based on their different structural features. They have differences in the structural features, but they are belonging to the same species, and these are called as serological differences, which can be determined, and that's why they are termed as serotypes. So actually we are talking about same species but 4 different strains. So Dan 1, Dan 2, Dan 3, and Dan 4. That is the reason why I'm talking about the four different serotypes. Now, when we are talking about the dengue or the dengue fever or the disease that we are calling it as dengue, is amongst the major tropical diseases in the world. Globally, also, that if you look at the epidemiological studies, it will clearly narrate that yes, dengue is one of the top 10 tropical diseases uh, that we should never neglect. That actually we should never neglect. And if you talk about the incidences, see, in if I tell you correctly, then in the past decade, the incidences of this dengue virus has grown exponentially. It has become into limelight. It has come into limelight in past 10 decades. Although it was there for for many decades that has been passed. But yes, in the past decade, in past 10 years, the incidences have grown up. The incidences have grown up. Why? Because in recent decades, alongside with the geographical expansion, this particular vector that I'm talking about, the vector that is Aedes mosquito, Aedes aegypti, that distribution of that vector has increased. That's why the dengue virus has gone globally. That's why it has gone global. So let us talk about what is dengue virus. When we are talking about the etiological part, I already told you that we have four different types of strains of this particular viruses. And when we are talking about the transmission, what is the vector of that transmission? Although majority of the places you will be seeing that Aedes aegypti, the Aedes mosquito is the main transmission vector of this particular disease. But apart from Aedes aegypti, albopictus, Aedes albopictus is also responsible for the transmission of this particular dengue virus. And apart from the mosquito bite, what can be the other routes of infection or routes of transmission in terms of when we are talking about the dengue virus spread? Then, of course, blood transfusion. If the blood is taken from an infected individual, then the individual blood cells will be containing the infected serotype, and then that blood, if given to a healthy individual, may result the onset of disease in the onset of disease, and will cause the dengue viral infection, or in cases of organ transplant. So till date, no case has been reported from person to person transmission. No case has been reported from person to person transmission. That means it never spreads from any kind of aerosolic or any kind of fluid. It always spreads from a vector, and the vector is mosquito. So the mosquito bite is the thing that you should be more worried in this scenario rather than person to person contact. And of course, in today's time, when we're talking about the social distancing and the physical distancing, there is no need of any kind of distancing in this particular dengue virus symptom. But that doesn't mean that it is harmless. That doesn't mean it is harmless. It is being caused by a bite of certain mosquito, and mosquito arises when there are unhygienic conditions. There is a stagnant water. So one should be very careful when we are talking about that particular type. So when I'm talking about the serotypes, I told you clearly that there are four serotypes. Now, each serotype, once you are infected with the particular serotype, you will be immune to that serotype for life. Each serotype will provide you specific lifetime immunity. The time span for that immunity may range from months to years. So what I meant by once you are infected with a serotype, I have four serotypes of dengue. Suppose I got infected with den 1. I got infected with den 1. So I took medication and I recovered. When I recovered, now I have the antibody repertoire and the memory repertoire in my immune cells, so that when next time this den 1 will be infecting me, I'll be able to mount the response and eliminate it. So what? I have a specific lifetime immunity against den 1, but that doesn't mean that I am immune to other three serotypes. If suppose after 6 months I got infected with den 2, I will again develop the major symptoms of dengue and I have to go for proper treatment. And that is the reason, in order to say that I am immune to dengue, you actually need to be infected with all the four serotypes. That should not happen. Don't take that statement as literal. That the infection should not happen, because anytime you are giving load to your body. But if immunity has to be attained, then in that scenario only immunity can be attained. But single infection with a single serotype will be giving you immunity against that particular specific serotype, not all the serotypes. So when we are talking about these serotypes, they have vast variations in terms of genotypes. they have vast variation in terms of genotypes, and all the serotypes are much more capable to cause this disease and take this disease to a fatal form. So when we are talking about the genetic variations, few of the genetic variations in each of the serotype, they appear to be more virulent and have greater epidemic potential. So one has to be very precaution about the spread of this particular dengue virus. And not even that, if you look at the sequence similarity, you will there is amazing studies that have been done to determine the common progenitor of these four serotypes, and they have speculated that the common progenitor may be 1,000 years back. And you will see that though they are belonging to the same species, these four strains are belonging to the same species, but the sequence similarity is around about 62 to 65% only. So the remarkable difference is of the order of 35%, 35 odd percent. So that difference, that much huge difference in a very small fragment of single strand RNA. What is the size? That is just 11 kb, 11 kilo base pairs. And in that short spread, if there is that much of differences, then of course those differences will be expressed in terms of phenotypic expression, and that expression comes as a structural part, and that difference in the structural part is seen as serological difference, and that's why we are calling them as four different serotypes. So I hope I made my point crystal clear. So let us go and study what are the structure, how that structure is going about. Although I said that they have different structural features, but then features and the composition is completely different narration. So what we are looking at a structure of dengue virus. Now, in that structure, you can clearly see that it's an enveloped virus, right? It's an enveloped virus. It is giving you a representation of a football, but don't mistake it. You cannot kick it. You cannot kick it. Even the immune cells sometimes fails to kick it out. So let us see what exactly it is. See, these surfaces, the yellow color resemblance, what you are seeing is actually an E protein, what we are calling it. So structurally, there are only three proteins which I am calling it as structural proteins. Remaining of the proteins that are coded by the genome are actually non-structural proteins. So among the structural group, we have three different proteins called as EMC. E protein that forms the surface which is present in the dimer and giving you a representation of a single stop. These are attached by a transmembrane protein, and by transmembrane I mean it is spanning the lipid membrane which is forming the envelope, and that particular protein is called as M protein. And beneath that M protein there is a capsid, the proteins which are actually responsible for forming the capsid, which is containing a single strand RNA genome, which contains a single strand RNA genome. So you see the single strand RNA genome is encapsulated in capsid, the capsid protein, and then the capsid is surrounded by a lipid envelope, and that lipid envelope contains two proteins. One is transmembrane protein, which is M protein, and another is the surface protein called as E protein, which is present in the dimer. So this is actually the simplest form of structural representation of the dengue virus. So as we know, what proteins are making that particular virus, and then let us go and see the remaining portion of that structural narration, the remaining portion of that structural type. See, when I'm talking about the remaining portion of the structural type, let us see here. See, so the spherical structure, what I'm talking about, is roughly around 40 to 50 nanometer in diameter, and as I said, that structurally on the surface we have three types of protein, ECM, but non-structural proteins there are 7 different non-structural proteins. So cumulatively, when I'm talking about the total proteins that are coded by viral RNA, the number is 10. There are 10 different proteins that are coded by this particular viral RNA, dengue viral RNA. Out of the 10, we are saying 7 are NSPs. NSPs means non-structural proteins, and the remaining 3 are structural protein. So based on that, this particular image, let us look at this particular image. See, this is a representation of a electron microscope. The white color representation, what you are seeing is actually the surface proteins, the E dimers, what we are representing, and this blue color are the antibodies that are used to neutralize them. So antibodies goes and binds to that particular E protein, and thereby they mediate its clearance. So this is a representation of an antibody and the viral protein which are bound. Antibody is bound to the viral protein. Fine. So these antibodies are shown in blue, whereas the white portion is, the proteins of the surface proteins. So antibody is doing what? Antibody is distorting the structure of the virus, the envelope. It is distorting the structure of the envelope protein. That's how it helps in neutralizing the this particular virus. So as per the complete structural information, if you want to have more details of the structural information, the complete structure is present in protein data bank. If the file reference number of 2R6P, the complete structure is present in that particular structural database. We all know that PDB is a protein data bank, which is a structural database, and then again, that particular file number I have shown, I've given you here. Then let us talk about the main part, that is the genome, the viral genome, how it is encoded. So this representation from 5' to 3' I have given you of a single strand RNA virus. Single strand RNA virus, which is a plus sense, it can directly be taken up by the ribosomes and to form a protein. Now you can clearly see here. See, for student, you should have a clear cut understanding that by seeing this picture, there is no intron, right? There is no intron. I told you 10 different proteins. Let us count. The left hand panel is for structural, the right hand panel is for non-structural proteins. We call them NSPs or NPs, depending upon different nomenclatures what people are adopting. Now let us talk about the structural protein. When in terms of structural proteins, there are three proteins: C, PRM, and E. PRM is premature M. So we'll be talking about that also in terms of the replication cycle. So 1, 2, 3, three structural proteins: 1, NS1, NS2A, NS2, NS3, NS4A, NS4B, NS5. So cumulatively, these are 7 proteins. So there is no intron. So when the ribosomes will be attaching to this particular RNA and they will be forming the proteins, there will be a polypeptide chain. That's why it gives rise to its collinear polyprotein. So if this E protein has to go on a surface, so it has to first get detached from that polypeptide which is formed from this particular RNA. It has to get detached, and likewise all the 10 proteins have to get separated in order to function individually. Otherwise, in a single peptide, they cannot function. Who will provide that particular function? See, if you want to cleave a peptide, what you need an enzyme that will cleave the protein, and that enzyme is called as protease. But if host protease, that means my protease, is able to cleave that protein, then it will completely cleave that protein, and that viral protein will be non-functional. And that is the reason why among these 10 proteins, the NS3, the yellow portion gene, codes for a protein which is actually a viral protease. It's a protease which is coded by the viral genome, and that protease is actually responsible for the cleavage of these proteins so that these proteins can function individually. So you see, the virus is bringing its own machinery, it is bringing its own protease, right? So let's talk about this particular segment, this particular RNA. The RNA is of 11 kilobase pair length, fine, and as I told you, it codes for 10 different proteins, and among that, two major proteins that I have highlighted here is NS3 and NS5. Why? Because these two are one of the amongst the largest of the proteins. Amongst the largest is NS5, of course. It is the NS5. Now that particular NS5 protein is responsible for the replication of this particular RNA. It's the principal RNA dependent RNA polymerase which is coded by this particular virus, the NS5. In fact, it is a multimeric protein, and it contains dual activity. It contains a methyl transferase which is responsible for the methylation, and as well as it contains the polymerase which is responsible for the formation of new RNAs molecules. And then the another protein that I'm talking about is NS3. Now this NS3 contains two activities. One is helicase. One activity is helicase and the other one is protease. So this NS3 protease activity is actually responsible for cleaving the polypeptide. So apart from these three, then what are the other functions that we are talking about, in terms of non-structural viral protein? Now the major function in order to uh, I have an army inside me which is very complex, and simultaneously on the other hand the army is very coordinated also. It has so many warriors that are sufficient enough to fight the enemy, to fight my enemy. What army I'm talking about? I'm talking about the immune system. And who is the enemy? Enemy is the virus. Enemy is the viral pathogen. I have enough ability. My army is one of the most aggressive, one of the most complicated, and one of the most coordinated army in the world. So if virus wants to infect me, it has to design a certain mechanism by which it can evade my coordinated immune response. And that is the function of this particular first protein, that is ns1. That is ns1. Now this particular ns1 protein, see, it is involved, although it is involved in early phase of replication, but it actually this particular protein actually activates our immune response. Fine. And not only activates, this ns1 has been documented very well, and it's been shown to be a major factor when we are talking about the vascular leakage that is associated in dengue, the vascular leakage that is associated in dengue. So this particular protein is responsible for mounting the immune responses. And then the proteins that are like ns4b, ns4b, this, and the last, that is ns5. This ns4b and ns5, these two proteins are responsible for inhibiting the major player of my body that will mount the antiviral response, and that player is interferon. So ns4b protein and non-structural 5 protein, these two cumulatively act to suppress the interferon beta, gamma, and alpha signal. So when the type 1 and type 2 interferons both are suppressed, so the antiviral state is not achieved, and then it is very easy for this virus to once infect a cell and then replicate and then infect the multiple cells, because those multiple cells, in those cells already we suppress the interferon signaling, because these two proteins never allowed the infected cell, the primary infected cell, to give a signal by releasing the interferon to the normal healthy cell. So they block the interferon signaling or the interferon production inside the infected cell. So that particular cell, infected cell, will not be able to sensitize the neighboring cells. Fine. Then apart from this, when I told you the ns3 function, the ns3 function is actually a helicase and a protease. So we understood the protease part, but what do you mean by helicase? Why you need helicase? See, helicase is required during replication. Helicase is required during replication in order to in order to give rise to sense RNA from an antisense RNA. It is required. Then and what about ns2? ns2a is also involved in replication, whereas the b, which is a very small hydrophobic protein of 14 kilodalton size, is actually acting as a cofactor for polymerase, is actually acting cofactor for a polymerase. So these are the listed functions which have been given in the lecture, that one should be having a very clear cut understanding that why these non-structural proteins are used. You see, these are the proteins which are mediating the replication of this particular virus inside the cell. So if I want to design a certain therapy, I want to design a certain putative drug which I can say is an antiviral, and that antiviral is responsible for suppressing the dengue infection, then what I am targeting? I am targeting these proteins, these non-structural proteins, as well as the structural proteins, yes, but these non-structural proteins, what I will target? If I want to suppress the replication of this particular virus, what should I target? I should target ns3. If I want that this virus should not be able to inhibit my immune system, so what should I target? I should target either ns5 or ns4b. So this is the main thing. Man, you have to understand the complete machinery what your enemy is bringing, right? So that's why the complete proteomic profile of this particular virus was taken out, and we now know that it contains the 10 proteins and what are the functions of those proteins. So we know the structure of these proteins also. That's why I showed you this particular image in the last slide. That's why I showed you the image. See, this is a three-dimensional structure, and the complete structure has been delineated. and it is present in the protein data bank as an entry, and we can use that particular information to design a potent antiviral against the dengue virus. So you see, when we have established that what are the functions of these particular proteins, now it's our job to narrate and understand clearly how this particular dengue virus is going for our application. Now I've taken this image from a wonderful review that has been published in Nature regarding the replication mode of this particular dengue virus. They have clearly mentioned every single thing what happens during that particular application process, so you need not to go anywhere. This particular slide is sufficient enough to suffice everything about dengue virus replication. So when we are talking about the genome, we already discussed that we have 10 different proteins. Of course, there will be untranslated regions there that is present in both the terminuses of 5' and 3' UTR. Now this will, when the virus is approaching to infect a cell, when a virus is approaching to infect a cell, the virus needs to gain entry inside the cell. In order to get that entry, it first has to attach. Now this dengue virus requires a receptor to attach onto the cell's membrane. So it depends upon ligand receptor binding. The ligand is nothing but the E protein which is present on the surface. So E protein is actually responsible for attaching the virus. So the attachment part is mediated by E protein. So if you want to inhibit the attachment by using a certain compound, you have to design a competitor of this E protein. Now this E protein and the other one should compete with themselves in order to bind to this particular receptor. So what are the receptors of this and this dengue virus? See, dengue virus, here we have shown you 3 different receptors. The first normal receptor present on a host is of course mannose receptor, MR. Then the DC sign is present on macrophages. Both the receptors we are talking about, the mannose receptor is specific for dengue virus, and the DC sign in the macrophage is a receptor for this particular dengue virus. Apart from it, there are FC receptors also, by which this particular virus can gain entry inside the cell. Now first let us look at this particular mediation. Now once this particular virus binds to a receptor, then the receptor mediated endocytosis process is happening. So both the receptors are responsible for bringing this particular virus inside the cell by forming an endosome, and this endosome will now be engulfing the receptor as well as the attached virus. So this particular endosome or the endosomal vesicle which is formed contains now the virus and the receptor. Simultaneously, the FC receptor. Now the FC receptor is specific for the constant fragment or the fragment crystallizable of an antibody. Now if the virus is gain entrance inside the host, so the antibodies which are specific for this particular E protein will be binding. The E protein will generate an immune response and the antibodies will be binding. Okay, the antibodies are bind. Then what? Now the constant portion of these particular antibodies can go and bind to these FC receptors. They can bind to these FC receptors. Thereby, they will provide an indirect attachment, the attachment to the membrane, and then the receptor-mediated endocytosis can be initiated, and then again an endosome will be formed. So it can gain entry by any of those 3 receptors. And once the endosome is formed, there is an efflux of the proton inside the lumen of the endosomal vesicle, which changes the pH, and this change in pH brings about the change in the conformation of the structure of the virus, the E protein and the M protein, and the nucleocapsid is released. Now the C protein which was forming the capsid along with the RNA is released inside the cytoplasm. So you see the change in the alteration in the pH. The alteration in the pH was sufficient enough for changing the structural integrity. The E and the M protein, they are conformationally stable on a defined pH. So what happens next? The alteration in pH released to the structural conformation change, and then these E protein fused with the membrane of the endosome, and then thereby they release that particular nucleocapsid. Once this nucleocapsid is released and the capsid protein is removed, the plus strand mRNA goes to the rough endoplasmic reticulum and attaches to the ribosome and attaches to a ribosome. Now what is happening? If it is attached to the ribosome, then what will happen? The first phase of translation starts. All the 10 proteins are now synthesized. Now, among those 10 proteins, there is a protease which will cleave the polypeptide, and the NS5, that is a polymerase, is now released. Now, that NS5 will act on this particular positive strand RNA. The NS5 will act on this particular positive strand RNA to give rise to an antisense negative strand RNA. And then it will utilize this negative sense RNA to make multiple copies of plus strand RNA. So you see, from plus to minus, and then from minus to multiple copies of the plus. This is how the replication is going about, mediated by the RNA dependent RNA polymerase, which is coded by the virus. Now, simultaneously, what is happening? Simultaneously, what is happening is not just that only the proteins are formed from the first origin strand. No, once these plus RNA strands are formed, then these are also used as a template for synthesizing the multiple copies of this protein, so that there are multiple units of each protein that are present in a single cell upon viral infection. And then the assembly starts. The C protein will be recognizing this particular single strand RNA. It will be forming the nucleocapsid. Then that nucleocapsid from the cytoplasm now will go to the endoplasmic reticulum. Why? Because the remaining proteins which are formed on the ribosome are present on the membrane of that endoplasmic reticulum. Now this virus will gain entry inside the rough endoplasmic reticulum, thereby the invagination will form an envelope around this particular nucleocapsid, and that envelope contains the lipid membrane of the RER, as well as the proteins which are present here. The proteins are which are present here. And what are those proteins? Those proteins are structural proteins. The three structural proteins. What are those three structural proteins? E, PRM, and C protein. The C protein has already formed the capsid, so the membrane contains only E and M protein. So once it enters inside the lumen of the rough endoplasmic reticulum, where the pH is 7.2, it just forms an envelope. There is no conformational change, and right now we are calling it as immature viral particle. So now what is happening? It is adopting the cell trafficking pathway. It will go from rough endoplasmic reticulum to Golgi body, and from Golgi body to outside. How it happens? It buds off and goes to Golgi body, where the pH is 6.7, the trans Golgi network. Now from this particular trans Golgi network, it will travel up and go to the last part, where the proton influx will be again happening, and the pH will be lowered to 6. And once the pH is lowered to 6, simultaneously there is an action of a protease called as furin protease, which will start cleaving and trimming the surface proteins. And then the vesicle will bud off, and the bud off vesicle, the pH will be around 5.7. And in this 5.7, now the rearrangement of this particular M protein starts. It releases the PR part, and now the M protein changes the conformation, giving rise to a mature virus. And that mature virus, surrounded in the endosome and in this bud, which is containing the membrane, now the membrane will fuse, and this mature virus will be released. Fully mature dengue viral particle is now released. So what is actually happening? If the viral particle is surrounded by the PR peptide, as in the case in the rough endoplasmic reticulum, we call it as non-infectious dengue viral particle. Whereas if half of the or some of the portions are removed, some of the PR peptides are removed from the E protein, we call it as partially mature. But at pH 5.7, when the complete PR peptide is removed and the E protein has gained the conformation of a football appearance, then we are calling it as the complete fully mature viral dengue virus. And that is the complete replication cycle mechanism, I should say, for the dengue virus. Now, apart from this, what is happening? What is actually happening here? See, we said that there is a protein called as NS1 protein. Now, that NS1 protein is formed inside the rough endoplasmic reticulum. It is released in the form of vesicle, and it goes and expresses itself on the surface of the plasma membrane. Now, once the dengue virus is infected, it it forms a protein, that is ns1, which is presented on the surface of the surface or the membrane of the infected cell, and this ns1 protein will now signal the immune system that I have arrived, now it's your job to remove me. And it acts as this ns1 protein is helping in turn helping us in determining that yes, now this cell, this host cell, this cell which belongs to me is now infected with a foreign virus. So multiple antibodies are available for detecting the presence based on the surface staining of this ns1 protein. Now, the things which we have discussed here inside the lysosome, because of the acidic environment, there's a conformational change in the protein, and that conformational change is actually pH dependent. That's the major part of this particular, from transition that we are talking about, from immature to mature transition, from immature to mature transition, and the complete structural part of these proteins has been illustrated and it is present in the literature form. So that is how the complete mechanism is actually working. So when we are talking about the polymerase, we know that now it is responsible for forming the new RNA strands based on the viral RNA. Now when we are talking about the helicase, what helicase is doing when there is no double strand, that what it is opening. See, helicase is required, to when there is a negative strand is used as a template to synthesize the positive strands, there is a bonding, and then it is required to detach this positive strand so that a new strand can be formed. Then we have methyl transferase. What it is doing, the methyl transferase, the virus methyl transferase will attach the methyl group. It will attach the methyl group on the newly formed RNA, thereby it prevents the RNA from degradation. And also it acts as a calling site for ribosome, so that ribosomes can recognize this particular RNA and go for the protein formation. And that is how all these non-structural proteins are contributing in a successful replication of the virus. Fine. Then let us go ahead and try to understand, after complete understanding of the structure, after complete understanding of the family, after complete understanding of the genome, the constituent genes, the constituent proteins, and the replication cycle, now it's our job to understand how this dengue virus is being transmitted. See, as I told you, it's an arbovirus, so it is transmitted from vectors, and those vectors are called as mosquitoes. And among those mosquitoes, the Aedes aegypti and Aedes albopicus is mostly responsible for spreading this particular virus. So let us take an example of Aedes aegypti. So what is happening, how it infects? Let us see the viremia. When the virus has crossed the barrier and is present inside the blood which is containing the virus load is called, the condition is called as viremia. So when we are talking about a 0 day and the first human has been infected and the symptoms have started, and the mosquito has taken a blood from an infected individual. It has taken the blood from an infected individual. Now, it after taking the blood, say after 5 to 6 days, it has bitten a normal healthy individual. Then after biting, then there will be an intrinsic incubation period of say 6 to 7 days, and then that particular second individual which was bit by the infected carrier mosquito will develop viremia, and that is how the disease is spreading. If there are 10 healthy individuals and Aedes aegypti, there will be no dengue. If there is one infected individual and there is Aedes aegypti, it will feed on the blood of the infected and will take the blood and will infect the other 9 in the vicinity. And that is how dengue continuously spreads. So what is happening here? Virus is transmitted from human inside the mosquito. Now see, the blood is being sucked by the mosquito. So while it is sucking the blood, the virus is never retained in the saliva. It goes directly to the midgut. Then the mosquito will infect a healthy individual. Then the virus will infect the blood cells or the lymphatic tissue, and then the virus will be released inside the blood, causing the viremia. Fine. And in that condition, when the mosquito bites the infected individual, the virus will go inside the midgut and starts replicating. and starts replicating. And because of the replication and increase in the viral load inside the midgut of the mosquito, it crosses the barrier and reaches into the blood, and from the blood it reaches to different organs of the mosquito. And among those organs, the organ which is responsible for transmission is salivary glands. So it affects the salivary glands. So you see, the affected mosquito will bite, and because the virus is now present in the salivary glands, then the virus gains the entry the normal host, inside the normal host. So this Aedes aegypti is primarily a daytime feeder. It never bites you at night. It's a daytime feeder. Fine, and it lays eggs and produces larva. Whenever you see a stagnant water that is being present in the immediate vicinity, you should be you should clearly be alarmed by that situation, so that the stagnancy can be removed, the stagnant water can be removed, and there will be no platform for Aedes aegypti to proliferate. So when we are talking about this during the mosquito feeding, when we are talking about the dengue virus, see, when the mosquito bites, the dengue virus is inoculated inside the dermis and the epidermis. Fine. Although sometimes what is happening, that certain because of a certain penetration, the virus can directly enter inside the blood, fine, inside the bloodstream. But yes, the inoculation is all most of the time it is either in the dermis or the epidermis part. And now, see, now what is happening? Because of that bite in the skin, this inoculum, or this delivery of the virus will elicit the immune response. And because our immune response is now elicited, the macrophages and the dendritic cells and the Langerhans cells will going towards the site of the infection. When they reach your site of the infection, fine, so they will take the virus and they will take it to the lymph node. That is their job. Fine. As soon as this virus reaches from the site of infection to the lymph node, it triggers the recruitment of macrophages. Now, when the macrophages are available, the multiple macrophages are available, I showed you in the replication cycle that those macrophages are containing DC sign. What is DC sign? DC sign is the receptor. Now, what you have done? You have served the receptor on a platter to this particular virus. You have served the receptor of macrophages on a platter in lymph node onto this particular virus. Now this particular virus will gain the access of the subsequent targets and will cause the dengue virus infection. As a result, the number and variety of the cells which a dengue virus infects increases exponentially. And then when the viral load is high, this particular virus can be disseminated to lymphatic system, where it completely infects the mononuclear lineage. Fine. So this is how the infection spreads, and after affecting the mononuclear lineage, it can cause infection in the splenic and the liver macrophages, thereby worsening the condition. So this is how the overview of the infection is going about, or this is how the transmission, after transmission, the pathogenesis goes about, how it infects the cells. So see, when we are talking about the dengue illness in WHO in 2009, in 2009, WHO clear-cut defined the classification. The classification for the purpose of improving, the clinical management, or the improving the how you treat about a disease in the clinical settings. So what they give you the warning signs. They give you the list of warning signs. What are those warning signs? Let us see. See, they give us the clear-cut warning signs that based on the criteria, if the patient has traveled in recent time, has traveled to a certain area where the dengue virus cases are very high, where the dengue virus cases are endemic in that particular area, and if this individual has traveled there, then the chances that he was bitten by Aedes aegypti and has acquired the dengue virus are very high. So he or she should not travel back immediately to their residence place. Why? Because what is happening, you are becoming a carrier of dengue virus and you are coming here, and if in your vicinity Aedes is available, then now you have become a source for that particular spread. Now the symptoms, when you're talking about what are the symptoms once the patient is infected, the fever will be there, The feel will be there, or either, then it will be followed by either of the nausea or the vomiting, the rashes and the pains will be there. Then truncate test is done, for determining whether the individual is affected with the dengue or not. It's very simple test. You know the machine which is sphygmometer, which is required for measuring the blood pressure. There's a green strap, and then the pressure is being pumped so that the strap goes tightens only, and you hold the pressure for a while, and then you release and open up the strap, and then you see the small, if the small red dots are coming, that is the positive indication of truncate test, and that means that yes, you might be getting affected by the dengue virus. Then it also induces leukopenia conditions. So laboratory confirmation can be done based on the RT-PCR test, and based on the antigen-dependent test, the laboratory confirmation can be done. But the WHO guideline, based on this criteria, divided the signs into two parts. First part is without warning, and the second part is with warning sign. So let us first focus on the with warning. So warning signs: the patient will be having the abdominal pain, there will be a fluid accumulation, there will be mucosal bleeding, there will be liver enlargement, and the enlargement will be very high, greater than the 2 centimeter. So all these signs, what they listed here, persistent vomiting, all these signs listed here give you a clear-cut indication that yes, the patient might be affected by a dengue virus. And then if not treated, if not clinically managed, that it can lead to severe dengue case. It can lead to severe dengue case. What is severe dengue will come in just a fraction of a minute. But what are without warning signs? Now, what they said, these signs, what we have mentioned here, these signs will never appear in the individuals, but they are still positive for dengue. And if not treated properly, they might develop in severe dengue also. So you see, the person might say that I don't have these many signs, so I am affected by dengue, not that much. But no, you got to have a proper treatment, because WHO has clearly given a guidelines that the severity of the dengue can come, and the dengue can be aggravated without any warning signs. And what is that severity what we are talking about? There will be plasma leakage. So once the leakage is there, then what is happening? There will be a shock syndrome, which is called as DSS, dengue shock syndrome. Once the shock syndrome is there, the leakage is there, then the fluid will start accumulating, and you will see that there will be a respiratory disorder, the respiratory distress will be there. And this particular bleeding, when we're talking about the plasma leakage, it will be very severe, and it can be detected by the clinician. And then in more terminal cases, the organ transplant is initiated, including the liver. So apart from this organ transplant on the liver, the major threat that is posed in the severe case is your impairment or dysregulation of CNS. It can lead to your unconsciousness, and if the severity increases, it can damage to your heart and the other organs, and eventually to mortality. So what are the factors that are involved? The factors what we are talking about are responsible actually for controlling this dengue virus disease response. How you can control this? What are the factors that can contribute to abrogate or to mitigate this particular dengue virus infection? So see, first of all, your immune status. If your immune status is very healthy, if your army is well equipped, if you have sufficient number of healthy cells, the immune repertoire is very good, then the immune response will be mounted against the infected dengue virus, and the severity of this symptom will not be aggravated. But what is happening? The macrophages which you have served on a platter in the lymph node will act as the repository for dengue virus, and they will utilize the macrophages and the monocytes to increase the viral load. Then it also depends upon the virus strains, what is the virulence of that particular virus strain, whether it can be neutralized, or that particular variation that we are talking about in terms of genotypic variation that has the potential to go for epidemic or not. Fine. Then apart from this, there are certain observations that have been made. Based on that observation, we are categorizing these factors. Now the observation, depending upon the age, the old age people, the old age people, and and the immunocompromised age, what we're talking about, where the immune system is going for deterioration. We're talking about the as soon as the age increases, the people go to the old age, then the immunity level goes down, and at that time the severity of this dengue viral infection goes exponentially high. And when you talk about children where the age is very less, then also the severity is very high. So you see, the children's and the old age people, where the age based immune system is somewhat not that efficient, that is the sign, that is the stage where the severity can arise. And not even the age, the genetic status. The studies have clearly demonstrated that the people with the dark complexion have been shown to be protected, more efficient in combating this particular dengue virus. So what are, based on this understanding, what are the probable targets? If I am a virologist, if I am a drug designer, if I am an ethnopharmacologist or immunologist, what targets I should be targeting? What targets should be in my mind while designing or while screening a putative antiviral compound? So, see, what are the targets? I told you in between the lectures also, I will be targeting the proteins which are coded by the virus, whether it is envelope, whether it is capsid or helicase or polymerase, whether it is M or E. Both can be used as a target. In fact, the E protein is an obvious target. Why? Because it is responsible for the attachment, and it is the protein which is present on the surface, right? So if I am using any antibody, it will be specific towards the E protein. If I am using any compound, that should be able to bind to the E protein, thereby change its conformation or deteriorate it. And if it is degraded, then the virus will not be able to attach. If there is no attachment, then there is no infection to the cell. There will be no replication of that particular virus. So out of the 7 natural NSPs, non-structural proteins, so two proteins that we discussed, 3 and 5, the helicase protease complex NS3 and the methyl transferase and polymerase complex of NS5, these two are the most putative target, because one is responsible for replication, another one is responsible for peptidal cleavage to give rise to functional proteins. And at both the stages, if you can use them as a putative target to develop the antiviral compounds. And not only the, when we are talking about the drug designing program, see, NS3 and NS2B were the first targets that were used for designing the antiviral products. And then NS5, when they recognize that NS5 is the principal polymerase, they use that as a potential target, and currently used as a target. But yes, apart from the antiviral, when we are talking about the vaccines, then the vaccines have been in 2015, I guess. Yeah, in 2015, the vaccine was approved for use in almost in, I guess, around 20 countries. It has been approved to be used in 20 countries, the vaccine, and it has shown very good results, very promising results. And currently it is being channelized and it is being used as curative therapy for this particular dengue viral infection. So in summary, what gist we have taken from this particular lecture is that once you get to know about the replication cycle, once you get to know about the players or the proteins that are coded by the virus, and then those viral proteins, at what stages they are acting, and which is the most feasible state for me to target in order to design a molecule which can eliminate the infection, that complete narration will come into play when you are able to understand the complete replication cycle based on the genome product of any virus. So let us go, as our tradition is always there, to summarize it with the MCQ so that we have a better understanding of a lecture. So which of the following are the examples of Flavivirus? So you have to understand, you have to first read what different pathogens are present in the Flavivirus. We have already seen the dengue virus, right? Whether the Japanese encephalitis or West Nile is there or not, it's up to you to answer. Then dengue virus code genome is of how much length? I told you about that. The severe cases of dengue virus is referred to what? We just had a brief discussion regarding that also. So dengue virus, when it gains entry inside the host cell, it gains entry by attachment. Then what happens next? What is the next stage? That you have to answer here. What is the effect of lysosomal pH? What is the effect of proton ions? Why the pH is so much important for this particular replication of dengue virus? Then as per the WHO estimate, how many cases of dengue infection occurs annually? When we are talking about WHO and the WHO science, then one should be aware what are the WHO reports that are released for this particular dengue virus, because those reports are based on a certain collection of data, and these are released every year, right? Then which vaccine strategies furthest? When we are talking about a putative vaccine and the strategies that are used, so it is whether the vaccine is of heat killed, or whether it is live attenuated, whether it is recombinant vaccine, whether it is a fusion protein that is being used as a vaccine. You can read it, because there is only one vaccine which was approved in 2015, and then I leave it up to you to identify what was the company, whether it was Pfizer, whether it was Sanofi Pfizer, or it is AstraZeneca, or any other. It's up to you to identify. I can tell you the year was 2015. Then how long does this protective immunity after getting infected lasts for dengue virus infection? Whether it is for days, whether it is for hours, or it can lead to up to years also. Then which protein can we expect on the surface of dengue virus infected cell? Which protein was getting infected on the cell which was eliciting the immune response, whether it was 1, 2, 3, 4? Then when we're talking about the vascular leakage, the plasma leakage, whether it is irreversible, once the leakage starts it will never stop, or it is reversible, we can stop that particular leakage, or it is always aggravated, or it can never happen. So it is up to you to answer these questions based on your understanding of this particular lecture. And for your assistance, I've given certain links for the articles which have beautifully summarized the complete articulation of this particular dengue virus, right from scratch of the genome, the different genome part, the immune responses, the structural part. They have summarized it all, so it is for your further reading, so that your curiosity can be substantiated by the amount of information that these papers have showcased beautifully. And then there is a WHO link also, the newsroom, what WHO offers for different viruses, and the detail what they have provided for dengue and the severe dengue cases, the dengue cases and the severe dengue cases, and the complete narration what they have given regarding the available treatment modules, that is also available in this particular reading. So students, this narration about dengue virus is now at the end. So I hope we are at the end or the last part. So I sincerely hope that you learned the structure, the replication mode, and the contribution that each viral protein is making just to complete the replication and to complete the life cycle. And based on that understanding, what targets we have defined, that also I sincerely hope that you have understood. So that is all from me today. I hope you enjoyed the lecture. So thank you so much. We will be again coming, we'll be again meeting in the next lecture with a different topic, and we will again look at the at one of the most pathogenic categories, that is virus. Till then, please stay safe, take care. Thank you so much.